Hello, welcome to my channel. Today we are going to learn about sterility test in pharmaceutical quality control, which is very important for confirming the sterilization process. Let us go in details. So sterility test is defined as testing that the product is free from viable microorganisms that the product is free from viable microorganisms. So this test is done in all the products wherever they specify sterility. So you will get injections, bulk solids for injection. Suppose, for example, drugs may be given in solid, then you have to reconstitute and take via injections. Such included including several materials for biological sources like heparin, hyaluronase, antibiotics, all they need to undergo. Even the syringes, needles, bandages, dressings, surgical instruments, catgut. Sterile water for injection, ophthalmic products, implants, uh, immunological products such as vaccines, serums, diagnostic preparations need to undergo sterility test. So this shows in pharma industry, whichever doing sterile products, you need to undergo this sterility test. It is very tedious process. Uh, Kindly take a care and listen to the class attentively. So now, see. So what are the precautions you need to take care? There are a lot of precautions. Few I am going to keep which are important here. So the sterility testing should be carried out in aseptic manufacturing technique. So whatever most stringent way you do, the environment is protected. Usually we tell in grade A room, or you can do the sterility test in grade B room, provided it has to be done in laminar airflow cabinet. Everyone know what do you mean by laminar airflow cabinet. What you need to do is that sterility test should be done grade A room, or grade B room can be done in laminar airflow cabinet. Now, the air in that room should be controlled. So it has to pass through HEPA filter. High efficient particulate air filters, they are established in pharma industry. Through this, air has to come into that room where we are doing the test. And pressure should be monitored. Any changes in the pressure should be notified with the help of alarm. And room should be airlocks. There should be only positive pressure. That means any air should not enter into the room. It should go out from that room on. So positive air pressure should be there. That is nothing but airlocks should be present. Then operator should be appropriately gowned. Sterile garments he has to wear. They need to be well trained and validated. The persons who are involved here, they need to be well trained and validated. Appropriate cleaning, sanitation, and disinfectant procedure should be taken place. It should be done according to the SOP of your industry. Frequently, the air quality should be tested. So that's what we are telling. Environmental monitoring need to be conducted. So this is the requirement. Still, let us see in some more in next slide. So this is the cabinet which we were telling. Laminar airflow cabinet. Uh, so, and one more important thing is that the the medium which we are using, uh, medium and the environment what we are using should uh, should help in the growth and multiplication of organisms or spores, etc. So whatever we are going to test, they should also help in growth of microorganisms. So we will see it. So one more thing you need to keep in mind, it is not possible to claim that a batch of product is sterile unless the entire content of each batch has been tested. So you can't test everything in the batch. So pharmacopoeias have given some standards how to select the sample, how many quantities you need to take in a batch. From that quantity, how many ml or how many milligrams need to be taken. They have already specified. We will go and learn them. So this is the diagram which we are telling about laminar cabinet or roomy. This is about filtration techniques. This diagram explains the filtration technique which is used during sterility process. We will learn in coming slides. So this is the introductory part of sterility test. Sterility test is done all the preparations which are claimed to have sterile. So this is the one thing. Now, what are the steps involved? There are mainly 5 steps in that entire process. You can figure out into the 5 steps. So first one is selection of the sample size. So as I was telling in previous slide, if you prepare 1,000 injections in a batch, you can't test everything there. You need to select some 4 randomly. There is specifications, we will see. Then selection of quantity of the product. So whatever the selected, 4 you selected, how many ml or milligrams you are going to select, that is the one which we are discussing. Next, we are checking about, learning about the media used. There are usually 3 medias used in the process that we will learn. Then method of testing. There are 2 methods how to test. Then finally, we are going to learn about observation and results. Observation and results. So this is the, in nutshell, what we are going to learn. So there are 5 steps involved. So first one, you can see it. Please try to listen. So here, selection of sample size. So in a batch, how many samples you need to select? See, parental, there are different preparations here as we discussed. So these are the things. Parental preparations, not more than, if the batch contains not more than 100 containers, not more than 100 containers. What is the minimum sample you need to take? 10% or 4 containers, which are great. So 100 samples you prepared, 10% means 10, that is greater. So you need to take 10 containers. So that is the what we are telling. Selection of the sample size, more than 100, between 100 to 500, between 100 to 500, you need to take 10 containers. They have told directly 10 containers. More than 500 containers, 2% or 20 containers, whichever is less. So in a batch of 1,000 containers, so you can take a 2%. Or 20 containers. That is, 20 containers will be less. Randomly, you need to select. You randomly, the person involved in quality control can select the samples. So entire 1,000 containers, we can't check it. That's what they have given. So next, large volume parenterals. So for example, sodium chloride, 1 liter. The sodium chloride 9%. So big containers here it will come. So any number. Here they didn't specify the containers as we discussed in the top. Whatever the quantity of the batch it may be, you need to take 20%, so 2% or 20 containers, whichever is less. Then antibiotic solids, many cephalosporins, augmentin injection, solbactam, many of the containers, which are you are giving, cefixime, which are they are giving in the form of solids, where they ask you to use sterile water for injection to constitute, or many of anti-cancer drugs. They also need to be reconstituted. For example, cisplatin or capecitabine or cyclophosphamide. Then all these will come under like these preparations here. See, bulk containers less than 5 grams. Bulk package, the total content of the drug, 5 grams, you can take 20 containers. You can take 20 containers. Greater than, greater than 5 grams, you can take 20 containers. Less than 5 grams, you can take 6 containers. Bulks and blends. See, bulk solids, we need to see here. Under this, we will see bulks and solids. We can see here solid product is more than what specified above 5 grams, more than 5 grams, then we need to go here. Ophthalmic preparations, non-injectables. For example, ophthalmic preparations, non-injectables, catgut, surgical sutures, veterinary uses. You can follow these requirements. Not more than 200 containers, 5%, 2 containers, whichever is greater. More than 200 containers, 10 containers. Single dose containers. Sometimes you will get single dose containers. So apply the scheme shown above as per parenteral preparations. So here you can see for parenteral preparations. So this is what we need to follow for this. If there is single containers, you need to follow this. So 2% or 5% or 2% or 5 packages which are greater, maximum total of 20 packages. So for bulk solids, bulk, then up to 4 containers. If there are 4 containers, you can take each container. So entire container you need to subject them. More than 4 and 50 to 20% are 4 containers which are greater. So here for large volumes, only less we are taking. Remaining all we are taking greater, whichever is greater than specified. So this is the quantity selection of the sample size. Next slide, we will study about the quantity of the product to be taken. So whatever you have selected, for example, here, liquid preparations. See, you taken 2%. Here you have taken large volume 2%. So 20 containers you have taken. Then what is the volume to be tested? You can see it here. So selection of the quantity of the product, you see, if liquids less than 1 ml, if the container having less than 1 ml, that means single dose containers, what we were discussing in previous slide, single dose containers, whole content, 1 ml entire we need to use for the test. 1 to 40 ml. Half of the container, half of the sample can be used. Greater than 40 ml, but less than 100 ml, you see, 20 ml you can subject for the test. Greater than 100 ml, what happens? 10% of the content you need to take. For antibiotic liquids, 1 ml. Irrespective whatever we are telling, 1 ml. For insoluble preparations, ointment, creams, and everything, you can see it if it is a suspensions. you can divide into solids and others. Here you can see it. Use each container provided not less than 200 milligrams. 200 milligrams. So like this, you need to select the quantity of the product. First one, we have selected the quantity of the sample size. We selected the sample size previously. Then quantity of the product, how much we need to take, we have selected. Then we are going to study, as we discussed, you see, we have discussed these two. We have discussed these two steps. We have discussed these two steps. Now we are going to study about the media. What is the media? Please try to concentrate. Now, what is the media here? There are three types of media which we can use. Fluid thioglycolate medium. In coming slide, I will tell the composition as well as which microorganisms can be assessed. Alternate thioglycolate medium, little advanced, little changes they done. Then soybean casein digest medium. Soybean casein digest medium. Some of the textbooks are telling without M, SCD. Both are the same one. Many of them are telling in the abbreviation of SCD or SCDM. M is nothing but a medium. Soybean casein digest medium. Once after preparing, there is composition. After that preparation, you need to sterilize the medium. Sterilization process is done by autoclave. Moist heat sterilization is best method of sterilizing the medium. If you sterilize by hot air oven, your medium will die. It will dry and smashed. Then if you medium by filtration, it cannot be done properly. Some of the nutrients can stay up on that end. If you sterilize by radiation, nutrients may go down. So the best method of sterilization of the medium is autoclave, that is moist heat sterilization. What is the temperature and pressure here? You see 121 degrees, 20 minutes, 15 lb pressure is mostly prescribed method for sterilizing the medium. What is the moist heat? Under the moist heat autoclave, 121 degrees, 20 minutes, 15 lb pressure. So this is the medium. So what are the compositions? See, the fluid thioglycolate medium. Fluid thioglycolate medium. It is mainly used to test aerobes, bacteria, anaerobes, and sometimes even microphiles. And this fluid thioglycolate medium used to examine clear liquids or water-soluble material. So whenever you are taking solid you dissolve in the medium specified. Then if it is clear, you can use fluid thioglycolate medium. The incubation temperature for the fluid thioglycolate medium is 30 to 35 degrees. 30 to 35 degrees we need to use. These are the various components or ingredients present in fluid thioglycolate medium. Agar, it gives, you can see it, ingredients, grams per liter. How many per liter we need to use? Then uses. Agar supports the growth of aerobes and anaerobes. Then just like agar medium for our antimicrobial activity. Cysteine lowers the oxidative and reduction potential, that is antioxidant property. Sodium chloride, salt, dextrose, food material, yeast, food material, pancreatic digest casein, source of nitrogen, carbon. Sodium thioglycolate is a reducing agent and neutralizing the toxic metals metals present in our preparations, or mercurial preservatives. Sometimes we also use preservatives. That also can be neutralized with this. Then resin sodium, it's an indicator. So this is the various medium, various composition. Now you can see it. One more is the alternate thioglycolate medium. What is the difference between the previous one and this is that it doesn't consist of agar and indicator which we told, rasazurin indicator. These indicators are not present in alternate thioglycolate medium. And one more important thing is that it is used to test turbid and viscous products. Previously, we were checking clear solution. Clear solution. Now here, this is used to check turbid or viscous preparations. And it is also, you see, it is also used to check anaerobes, the bacteria which can survive in absence of oxygen. Previous one, both. Both. So these are the ingredients. Now, let us see next. Soybean casein method. Soybean casein. Soybean casein digest medium. It is also used for turbid and viscous methods. One important thing is that you can check fungi. Remaining aerobic bacteria, you can check. You can check. This is soybean casein. You will be having soybean as ingredient. Pancreatic digest, see the source of amino acid. Dipotassium hydrogen phosphate, oxidative reduction. Previous mediums was having cysteine. Here they are using dipotassium hydrogen phosphate. So these are the ingredients. What is the use of soybean casein? It is used to test fungi and the preparations which are turbid and viscous. Viscous. So this is about the medium preparation. So now one more test, important test. So before doing actual test, we want to check whether the medium is supporting the growth of microorganisms or not. So this test is called as growth promotion test, otherwise called as media validation. Whatever you prepared media, what I told you, the environment should be clean, but we want to check whether the preparations are free from microorganisms. So how can you do that with the help of media? And the media should also help in growth of microorganisms. If microorganisms are present in sterile products, and in order to check that, this media should help to growth. So that's why we do growth promotion test. Growth promotion test. So this test used to check the media whether it supports the growth of microorganisms or not. How to do? You take 100 ml colony forming units, 100 colony forming units, whatever the bacteria you selected, and add into the media, which are we told. If it is fluid thioglycolate medium or alternate thioglycolate medium or soybean casein digest medium, any of the medium you can select depending upon your product, and to that you add. Within uh, within 3 days, for bacteria, you need to show that there is some growth. For fungi, what happens is that you need to check for 5 days. Within 5 days, you need to have some growth. So this is the growth promotion test which we do for this end. So these are the some of the bacteria or fungi which we use. So what are those, you see? If you are using fluid thioglycolate medium, as we told, it is used for both aerobic and anaerobic. You can use Staphylococcus aureus or Clostridium sporogenosa or Plasmodium aeruginosa. So incubation temperature is given. So alternate thioglycolate medium. You can see Clostridium sporogynosa, you can use the best microorganism, and Bacillus subtilis for aerobic. Then soybean casein digest, Aspergillus, Brasiliensis, and Candida albensis for fungi. You can use soybean casein digest medium. So here you can observe the incubation temperature is different from the previous, and here for this soybean casein digest, the incubation temperature is 20 to 25 degrees. So these are the test microorganisms. Now, this is one important slide. So these are the some of the, suppose if the preparation itself is antibacterial, or if you use some preservatives, then that will inhibit the growth of microorganisms. So to nullify that, for example, you are checking penicillin, ampicillin, amoxicillin injection, for example, cephalosporin, any injection you want to check. But antibiotic itself will kill the microorganisms. So what you need to do is that you need to nullify them, that antimicrobial activity, and help in the checking the true sterility process. So if there is penicillins, you can use penicillinase. So this penicillinase will nullify the penicillin. If the cephalosporins are there, cephalosporinase. For example, sulfonamides are there, you can use PABA. So this para-aminobenzoic acid will nullify the sulfonamide present in it. We are only checking the product, whether it is sterile or not. Suppose if the products have halogens, you can use sodium thiosulfate. So if the product has alcohol, you can dilute to such an extent that alcohol concentration will be less than 1%. If apart from this, if any one of the you have, so if there is another antibiotics, you can filter by membrane filter. So all the antibiotics will stay up, only product, pure preparation will come in. So these are the some of the things. That's what you see. Inactivation of antimicrobial agents in sterility test. Why we need to do? So this will give false results. So we make them inactive. Here, sterility test, we are not checking the potency of the preparation. We are checking whether the product is free from microorganisms, that is viable microorganisms or not. That's why we use this. We use this. So please try to concentrate. So almost we are going to end. So test methods. So there are two test methods. Test method is membrane filtration method and indirect inoculation method. You can select your method, but most preferred method is, according to pharmacopoeias they prescribe, is membrane filtration method. Direct inoculation method, you feel it is very easy, but you need to have expert. So membrane filtration method, what is this? You take the sample, whatever the specified, whatever we learned in step 1 and step 2. So you need to filter with the help of membrane filter. So allow the sample to fall. Now you take this filter paper, cut into 2 and place it in fluid thioglycolate medium or soybean casein digest medium. Any of the medium you can in the medium, and check whether the microorganisms are grown or not. Why? We are not using the product here. See, we are making the product to pass through the filter paper. Now the filter paper is cut here. The filter paper is cut. So the whatever the contents is present, it came here. It came onto the filter paper. Now that filter paper you are checking. You can see it here, 0.45 microns filter paper we use. The flow rate is between 55 to 75 ml at the pressure of 70 mm of Hg. Usually we take cellulose nitrate filter papers are used. Now, once the filter paper is cut, that you are placing in that media and incubating it for 14 days to check whether the microorganisms are grown or not. Within 14 days, if there is no microorganisms are grown, then the product is past the test. So what are the various products which can be sterilized by filtration methods is oils, oily preparations, for example, vitamin K, all vitamins. They also need to undergo sterility test because they are injections, no? So ointments, which are sterile, soluble powders, liquid products, non-bacteriostatic solids. All these can be done by membrane filtration method. Next one, we can see it, direct inoculation method. What happens is that you dissolve the sample in the medium as specified in pharmacopoeia. You take a sample through injection, 1 ml or 10 ml, whatever we have told. Then add on to the fluid thioglycolate medium or soybean casein digest medium. So how much we need to add? Not more than 10%. Then incubate it for 14 days and check whether the microorganisms are growing or not. So these are the sample preparations which can be used for direct inoculation method. Generally, they are telling this is very tedious process. Expert should be presented. So some of the preparation aqueous solutions can we undergo, suspension they do, oils, oily solutions, ointment, solid, medical devices, all can be tested. How can we do medical devices? So for example, if the syringe or injection, you take the sample, that syringe you dissolve in a solvent, and that solvent is subjected for test. Suppose if it is cotton, you can take the sample specified in the cotton and just place it in the mediums. If it is gadget, you can do the same thing. Suppose if it is medical devices, syringes or IV setup, then how to do it is that those devices you need to dissolve, you need to place it in a solvent as specified in pharmacopoeia, and that solvent is subjected for any of these two preparations, any of these two methods, that is filtration method or indirect inoculation method, direct inoculation method. So these are the various methods. Then finish almost. So results and interpretation. How to do these results and no. So I'm telling that. So incubation period is 14 days, whereas media validation incubation period is 3 and 5 days. 5 for fungi, 3 for bacteria. So here is results interpretation. You see, the sample passes after 14 days. The sample passes the sterility test if there is no growth of microorganisms after incubation. Now, if the growth is observed, or turbidity is seen, you are not able to observe clearly. You are not you have a doubt whether it has growth is there or not. Then you need to retest it. If the growth is observed again in the second test, then sample failed. So you can see if the growth observed again in the second test and cannot distinguish from the first test, you are not able to distinguish between the first test and second test, then also it fails. Then also the test is called as the samples are not sterile and it is failed. Now, but if you can distinguish from the first test, telling that second test is less than from the first test, then what happens is that you see, then, but it can be distinguished from the first test. Then the second retest is done twice the number of the samples. Again, again you are doing twice the number of the samples. So here, what is the criteria? If directly passed, no growth, no issue. Second one is that if it doesn't pass, you found some turbidity or some growth and you are not sure, then what happens? You are repeating the test and you are checking, comparing with the old one, with the old one. If there is any disturbances, any less or more, if you can see it, more I can't tell if you, but if can be distinguished, less generally it will be less. Then what happens? You are retesting it twice the number of the sample. If now in this, if the growth is not observed in the second retest, it passes. If growth is observed, then the test is failed. So this is about the interpretation. Again, I will tell first one direct. Second one is that if you observe some growth or you are not able to distinguish, then you need to retest it. Retest. If there is any change, you are doing again with the double samples. Now on the second test, there should not be any growth. And the second test, what we are retesting by double the quantity, you should not have any growth. If there is growth, that means the product is not sterile and it should be rejected. And QC department will not give approval for the marketing. So this is about sterility test. Thank you. If you have any doubt, you can comment in the comment section. We will reply. Thank you very much.